In this video, we will explore what genomics is, the applications it has to fisheries and conservation management, and what steps managers and resource users need to take to implement genomics into their practice. Genomics is the study of all the DNA in an organism, called the genome, and this is how genetic information works together to shape traits, health, and responses to the environment. Genomics is especially useful in fisheries and conservation because it can help identify species, reveal population structure, and address a wide range of stock-related questions. These insights are critical for understanding how fish populations are connected, how they're changing over time, and how best to manage them sustainably. When looking at a population of fish, whether captive or wild, it can be difficult to determine what is truly happening beneath the surface. Fish populations may appear to be healthy or uniform, but important population structures and dynamics may not be visible based on appearance and abundance alone. For example, genomics can reveal whether a population has high genetic diversity. High diversity often indicates a stable and resilient population that is better able to cope and adapt to environmental change. Conversely, low genetic diversity may signal inbreeding or vulnerability to stressors. Genomics can also be used to assess intergression between captive and wild fish. This allows managers to determine whether hybrid individuals are present and how much genetic mixing is occurring. This information is critical for protecting wild populations and for maintaining genetic integrity. In some cases, genomics reveals that a species once thought to be a single population is actually composed of multiple distinct populations. This discovery can require a reevaluation of management unit boundaries and stock assessments to ensure each population is managed appropriately. Recognizing these differences is essential for effective conservation and long-term sustainability. Collecting genetic information is relatively straightforward, and this can be done in a minimally invasive manner. This typically involves taking a small tissue sample, such as a fin clip, and preserving it in ethanol. These samples can then be analyzed to generate genomic data that support management and conservation efforts. So what does genetic data actually look like? Well, here is an example of genetic data that was used to assess stock structure. Each bar represents an individual fish, and the colors here show genetic ancestry. Similar patterns indicate shared stocks, while differences reveal hidden population structure. Overall, genomics has countless applications including species identification, population connectivity, stock structure, hybridization, conservation planning, and many others. Integration of these tools can serve to provide managers with more precise information, leading to better informed decisions. As genomics tools continue to advance, integrating them into fisheries management will become increasingly important for protecting aquatic resources in a changing world. To learn more about the use of omics in Canadian freshwater fisheries and conservation management, and for access to our database, tools, and resources, be sure to visit genfish.ca and click on the Omics Applications in Fisheries panel, or simply scan the QR code on the screen. Finally, we ask you to take a moment of your time to scan this QR code and complete our short Omics survey.